Okay, chapter 9, amplification of nucleic acid. So the PCR, all right, so the polymerase chain reactions, all right, it's a primer-directed in vitro enzymatic reaction to amplify a defined DNA region. Okay, so primer-directed, so that means during the PCR reaction, you need the primer. Primer basically it's a short oligo fragment, like a 10 to 30, 18 to 30 nucleotide long, all right? And it should be a pair because the primer would define the region of the DNA to be amplified, all right? So that's why it's called primer directed, okay? Because for this pair of a primer, they can define the area that to amplify. It's sort of like the in vitro DNA replication, DNA synthesis reaction, all right, and enzymatic reaction. So during this reaction, when we discuss the replication, so you know that we need a DNA polymerase, all right, to perform the DNA synthesis. So for the in vitro reaction, again, so you need to have this DNA polymerase, all right? So that's why it's an enzymatic reaction. And for the production of a specific DNA fragment, because the primer define this specific region to be amplified. Okay, so that's the PCR, polymerase chain reaction. So normally you need a template, right? The template DNA, and then the first step you need to denature this double helix DNA so that this primer will be able to find a target and then adhere to the target DNA sequence. Once they adhere to the target DNA sequence, then they can perform the DNA synthesis, right, to amplify the DNA, right? So that's one cycle, and then you can repeat these 3 steps again and again for 20 to 30 cycles or 40 cycles, so that you can amplify many amplicon. We call it amplicon. All right. Every cycle will give you. So, for example, originally you have 1 double helix, right? And then that's 0 cycle, 2 to the power of 0, so it's 1. Then after 1 cycle, then you have 2 pairs. All right, 2 DNA double helix. Therefore, it's 2 to the power of 1. So it's 2, all right, after 1 cycle. After 2 cycles, 2 to the power of 2, that would be 4, right? So that's a 2 to the power of n amplicons, depending on the number of cycles. So that's a PCR reaction. Okay, so the primers, all right? So primer is very important because primer would define where to amplify, to define the region where to amplify, and later on when we yes, I think we have that information that we would discuss about the COVID-19 amplification. So basically, the primer, so that can define the region to amplify. So that means the first step before you design the primer, you need to know the DNA sequence. All right, so that's the reason why when you have this COVID-19, the SARS-CoV-2 virus, the first thing you need to do is to determine the genome sequence, because once you determine the genome sequence, then you will be able to design the PCR primer so that you can perform the test to determine the presence of the virus. All right, or even use the genome sequence to design the vaccine. So like a Moderna vaccine or the Pfizer vaccine, they have this messenger RNA vaccine. All this is not possible unless you have the genome sequence. Okay, so for PCR again, the same. The only thing that you need for the PCR reaction is you need to have this DNA sequence, and so that it should be non-DNA sequence, so that you'll be able to design a primer, okay, and also the distance between the primer binding site that can determine the length and the size of the PCR product, because the distance here that will determine the size of the PCR product. Okay, and the primer complementary to the sequence flanking to the region to be amplified, right? So that's PCR primer. And then to perform the PCR reaction, of course, you mix the DNA template, DNA primer, and polymerase, and also you have to supply the deoxynucleotide, because during the DNA synthesis you need the supply of the deoxynucleotide, and then so that they can perform the DNA synthesis. Okay, and this is a typical protocol, the ingredient that you need to use to set up the PCR reaction. So for example, for the primer, it normally is about the 250 nanomole of each primer, and then 200 nanomole of each deoxynucleotide. All right, and then you have the salt and magnesium. All right, is a cofactor to promote the enzyme activity. And then you need to have polymerase, okay, and template, and so that you can perform the PCR reaction. Okay. So in the typical PCR reaction, each cycle, as I mentioned earlier, 3 steps. The first step, you need to denature the double helix DNA. And to denature the double helix DNA template, so normally you have to increase the temperature to the 94 degree. All right, so remember 94 degree, very high temperature. At this temperature, normally all the protein will be denatured. Right, now we learned previously, protein will be denatured at 94 degree, and unfortunately, the polymerase, DNA polymerase, is enzyme. It's a protein, so all the DNA polymerase will be denatured at this temperature. Therefore, for the PCR reaction, it is important to use a very specific, unique DNA polymerase, and this DNA polymerase, we call it a Taq polymerase. The Taq polymerase is purified from a thermal bacteria that are found in hot spring, so that this special Taq DNA polymerase, okay, they will be stable at this high temperature. Okay, so that's the only thing that you need to know, very specific, that special DNA polymerase to be used for the PCR reaction. So you need to have the denaturation, and then once you denature the double helix DNA, then you cool down the temperature to 50 to 60 degrees. This depends on the annealing, the melting temperature. Okay, so the annealing temperature will be normally we set up at 2 degrees below the melting temperature. That we will discuss further later. I think we mentioned this in the chapter 1, the melting temperature. That will be in the population of a DNA molecule at the temperature where 50% of population is the single-stranded DNA, and the other 50% of the population is double-stranded DNA, and that temperature we call it a melting temperature. All right, so normally we set the annealing temperature is 2 degrees below the melting temperature, so that since we set up 2 degrees below the melting temperature, that would suggest more than 50% of the population will be double helix. All right, so when they cool down from 94 to 50, all right, so more than 50% of primer will find their target, all right, and then bind to the target sequence, and then increase the temperature to 70 degrees, which will be the optimal temperature for the Taq DNA polymerase, and then they can perform the DNA synthesis. Okay, so that's the 3 steps, and that's one cycle. And here is just the simplified the formula to calculate the melting temperature. So the melting temperature depends on the number of the G, C, A, and T in the primer. So for example, in this primer, so you have 4G, 5C, 7A, and 1T, right? And then the melting temperature will be 4 degrees multiply the number of G plus C, which is 9, plus 2 multiply the number of A and T in the primer. So that'll be 7 plus 1 is 8. So that'll be 4 multiply 9 plus 2 by 8. So that'll be 52 degrees. So this is the simplified the formula, the quick formula to determine the melting temperature. So that in this case, then the annealing temperature you will set up as 52 minus 2, 50 degrees. So that to ensure more than 50% of the primer will bind to the template. So that's the how you determine the melting temperature and the annealing temperature. And to be more specific, for the first 3 cycles of the PCR reaction, okay? And so this is the first cycle. So you have the DNA template and then you denature the DNA template, right? So you have this long product will be produced, because once you separate these 2 templates, then the primer bind one end and extend to all the way to the other side. So it's a long product. And then we call this as the forward primer, reverse primer, then the amplify all the way to the end. So you produce these 2 long product, all right, in the end of first cycle. Then when you enter to the second cycle, so you have this long product, okay? So this green one, that'll be the original template, and the green one in the cycle 2, they will just replicate the same long product as the cycle 1. And we focus on this inner 2 product here. So with this long product from the cycle 1, then at the cycle 2, this forward primer bind to this location. Then when they extend it, they will only extend to here. So that will give you only the short product. And for this one, reverse primer bind here and then extend it to here, all right? And that will give you the short product. So after the cycle 2, then you will produce this 1 short product and this 1 short product, okay? And these 2 will be the same as cycle 1. And since you produce this short product, then when we enter into the cycle 3, this will be the same as the cycle 1, all right? And this long product, all right, cycle 1, and this one will be the same as the cycle 2. And now, if we look at this short product, okay, these 2 short products, they will only amplify the exact size of the PCR fragment, okay, your target PCR product, okay. Only this short product from the cycle 2 will give you the exact product that you want. So that's the detail of the first 3 cycles of the PCR reaction. Okay, any questions so far? The whole class. Very good, Aidan. Okay, so now again, so the amplicon, the number of amplicon, okay, so it's 2 to the power of n, all right? N is the number of cycle. So that's the number of the PCR product. And then, so the PCR, the reaction, as you can see, we have to repeat those 3 steps for 20 times to 30 times, right? Each time we call 1 cycle. So that we need to repeat the temperature change. And since it'd be very inconvenient to use, let's say, 3 different incubator with 3 different temperature, and then you switch from 1 incubator to another incubator. That's not convenient. Therefore, the field, they develop this machine. It's called a thermocycler. So the thermocycler, they can change the temperature in a block or chamber, and that you hold a sample, all right? So that would be specific equipment, all right? We have many of these PCR equipment in the laboratory, in the department, all right? And also later on when we discuss the real-time PCR, my laboratory also have a real-time PCR equipment that can perform the real-time PCR analysis. And now this is just the regular PCR equipment. And I mentioned that for the PCR we need a polymerase. Okay, the polymerase is one of a DNA manipulating enzyme because it can make copies of molecule. So as we learned previously, three different DNA polymerase. The POL3 will initiate the DNA replication in the beginning, and the POL1, that's when in the end of the or during the replication. So when you degrade those RNA primer, you need a POL1 to fill in the gap, right? And then the POL1, right? POL1, DNA polymerase 1, not only to fill in the gap, but also to replace the original nucleotide. That's because the POL1 has the exonuclease activity, 5' to 3' exonuclease activity, so they can degrade those original existing nucleotide and then add a new nucleotide. And then clinone, that's part of a POL1, right? That's part of polymerase 1, that's clinone. And the clinone, basically, they can only fill in the gap because the clinone does not have the exonuclease activity. And then also another two enzymes, reverse transcriptase and Taq DNA polymerase. Taq DNA polymerase, as I mentioned, the one that we commonly use in the PCR reaction because the heat stable DNA polymerase. And reverse transcriptase, basically, they use the RNA as a template to synthesize the complementary DNA. So that's a reverse transcriptase. Okay. So here's the detailed description of POL1, right? Polymerase and exonuclease activity, right? And then when you digest the POL1, you get two different polypeptide. One part is called clinone, right? So the clinone does not have 5' to 3', this 5' to 3' activity, because here, if you don't have a 5' to 3' activity, then you cannot replace the existing nucleotide. Okay, but the clinone has a 3' to 5' activity, so that they can check whether the nucleotide, whether they incorporate the correct nucleotide. If it's the wrong one, they can still remove it and put the correct one. So that's 3' to 5' exonuclease activity. Okay, so you can repair the mismatch. That's clinone fragment. This 5' to 3', that's only for the POL1, because the POL1 can replace, when they can replace the existing nucleotide and then put in the new nucleotide. And then thermostable polymerase, that Taq DNA polymerase, right, purified from the thermocell particles, bacteria. Okay, that's commonly used in the PCR reaction. And then to evaluate whether PCR reaction is successful, all right, we have to examine the PCR product. And then the simplest and the easiest procedure is to analyze the PCR product by agarose gel electrophoresis. Okay, to do the agarose gel electrophoresis, so that normally the sufficient DNA have been produced through the PCR reaction, so that the DNA fragment should be visible under the agarose gel electrophoresis. So after the agarose gel electrophoresis, you can even find the correct PCR product and then purify the product directly from the agarose gel, so that you can have two birds in one stone. Not only to evaluate the PCR product purity and the quality in quantity, and also you can purify directly from the gel. Okay, so that's the agarose gel electrophoresis to evaluate the PCR product. And then to avoid the misprime, so the misprime, so for example here you will see this extra band. This is correct PCR product, and then you can see this extra band, right? This extra band, that can be the misprime, right? And in the bottom here, that's a piece primer dimer. But this extra band here, that will be misprime. Okay, to avoid the misprime, there are several ways, because misprime occur due to non-specific hybridization of primer. So when will this happen? If we go back to here, if you look at these, the binding of primer to the template, all right? So as I mentioned, in order to increase the PCR, the efficiency, so we set up at 2 degrees below the melting temperature. So that means you have more than 50% of the population is the double helix DNA. If you see the misprime product, normally you will need to increase annealing temperature. All right. When you increase annealing temperature, that will reduce the binding of the primer to the template. So that means that will reduce the non-specific binding. Only a very accurate complementary DNA sequence will form the double helix. All right, so that's one way to avoid the misprime. Okay, to increase the annealing temperature. All right, so that's why you have to use proper annealing temperature. And then you can also design primer carefully, because the primer you design, let's say 18 to 20 nucleotide long, it might have 50% or more or 70% similarity to other sequence in the template. So normally when you design a primer, you will double check whether that they have homology to the other region of the template. All right, so you will design the primer carefully. And however, there are several online software available. They've been designed so that you can put in your template sequence, and they can find the best available primer sequence for you. So that's the primer. Design primer carefully, and then adjust the monovalent cation concentration. Use the hot start. Prepare reaction mix on ice. That's the standard procedure. You always prepare the PCR mix on ice, all right, before you start a reaction. And always add the enzyme the last step, right before you put the reaction into the thermocycler. Okay, that's how you avoid the misprime. And then the product cleanup. So as I mentioned, if you perform the agarose gel electrophoresis, then you can purify the product from the gel directly, the gel elution. Or you can do the solid phase isolation of PCR product, or DNA precipitation. That what you can do is to add the ethanol and the salt, so that you can precipitate the DNA in cold temperature. And addition of this SAP, the stream alkaline phosphatase, in combination with exonuclease, that can remove the nucleotide and primer from the PCR product prior to the sequencing. All right, that's SAP. And the contamination control. So normally you design this pre-PCR area and the post PCR area, that's a physical separation, right? Physical separation. Or you can add this deoxy-UTP and the uracil and glycosylase to your PCR product, to the PCR reaction, okay? Because this will involve the substitution of the deoxy-UTP for the DTTP in the PCR reagent master mix. And so that the result is you will incorporate the deoxy-UTP instead of DTTP to the PCR product. All right, so at the beginning of each PCR reaction, the enzyme uracil and glycosylase, that's UNG, all right, it's called UNG. You can add it to the reaction mix so that they can degrade any nucleic acid containing a uracil, like the contaminating a PCR product from the previous reaction. Okay, so that's the UNG. Or the sorolin. So you add to the amplification product and plus the UV, all right? So the sorolin intercollect between the bases of double-stranded DNA and in the presence of a long-wave UV light. So they covalently attach to the thymidine, uracil, and the cysteine in the DNA chain, and then the bucket adduct of the sorolin prevent the denaturation and the amplification of the treated DNA. Okay, that's how you prevent the contamination, or you can use the 10% bleach for the surface decontamination. So these are the contamination control for the PCR. And of course, there are many different applications, right? And now we will discuss the modified nucleic acid amplification technique. All right, so there are different modifications, and we will discuss this nested PCR, multiplex PCR, reverse transcriptase PCR, digital PCR, and the quantitative real-time PCR. Okay, so for the reverse transcriptase PCR, okay, it's called RT-PCR. So you normally see the term as qRT-PCR. Q mean real time, all right? RT does not mean real time. RT mean reverse transcriptase, okay? So qRT-PCR mean real time reverse transcriptase PCR. qRT-PCR basically is the only protocol nowadays in the world that they use to determine the presence of the SARS-CoV-2 virus in your body, okay? So it's qRT-PCR. So first step, you have to perform the reverse transcriptase PCR and then perform the real-time PCR. So it's a combination of both. That's why it's called qRT-PCR, to determine the nowadays during this pandemic time, to determine the presence of SARS-CoV-2 in the human body. Okay. So the reverse transcriptase PCR basically they convert the RNA to double-stranded DNA. Okay. So normally you have this oligo-DT primer, which is 18 base long single-stranded poly-DT sequence that will prime, right, to the RNA, right, and then they can synthesize this complementary DNA. After they synthesize the complementary DNA, you have this the hybrid RNA and the DNA hybrid. So at least this time you need to degrade this RNA. All right, degrade this RNA. So you have different methods. So one of the commonly used method is to use the RNSH to degrade the RNA, so that you have this cDNA, single-stranded cDNA, and then you can synthesize this double-stranded DNA, and then you can amplify the double-stranded DNA from here. Okay. So oligo-DT primer, okay, that's one way that you can synthesize RNA, or you can use the random hexamer or decamers, which are the 6 or 10 base long single-stranded oligonucleotide random sequence. Okay, this primer will match and hybrid to random site in the target RNA to prime the cDNA synthesis. And this primer, they can generate the cDNA from all RNA in the specimen. And then the last step, of course, is just amplification of the cDNA by PCR. So this is a reverse transcriptase PCR. And the nested PCR was developed to increase both the sensitivity and the specificity of PCR, because this technique used 2 pairs of amplification primer and 2 rounds of PCR. You need to perform the PCR 2 rounds, okay? And then because they have 2 pairs, all right, 2 pairs of the primer. So you have the first round, use the first pair, and then you take the product from the first round and apply the second pair, and then to proceed for the second round, okay? And so that they can increase the sensitivity. And the increase the sensitivity arises from the high total cycle numbers. And the increase the specificity arises from the annealing of the second primer set to the sequences produced by the first round. All right, so remember how they increase the sensitivity and the specificity. And the only major concern for this method is the contamination that occur during the transfer of the first round product to the second tube for the second round of amplification. Okay, that's the only concern, the major concern, because you have to perform these 2 different rounds. So you have to transfer the product from the first round to the second round. Then you might introduce the contamination. Okay, so the traditional nested PCR, so that you have two set of primer. One set is outside, all right, and then the second set of primer will be inside the product from the first primer. Okay, that's the traditional nested PCR. Then they have the modified nested PCR. Okay, it's a semi-nested asymmetrical PCR. So that means you have 1 primer that can be used 2 times. Right? See the blue one here on the left. You can use it and then match with this, coupled with this pink one, the outside one here. So that will be the first round. And then the second round, you use the same blue one, but the different pink one. Okay, so that's called semi-nested PCR. All right, any question? Very good, Crystal, D, and Yasmin. Good. So now these are the reverse transcriptase and nested PCR. So let's watch a short video, okay, before we continue, so that we learn a little bit more.
What is a normal nested PCR is based on a standard PCR reaction. PCR is a technique used to make multiple copies of a DNA fragment through repeated cycles of strands separating and replicating. PCR is based on the natural process of DNA replication and has 3 steps: denaturation, annealing, and elongation. What is nested PCR? For nested PCR, 2 sets of PCR primers are used to amplify a specific DNA fragment. The first pair of primers amplify a fragment similar to a standard PCR. The second pair of primers, the nested primers, bind within the first PCR product.
Outer and inner, right? That's nested.
When using nested PCR, a commonly occurring problem with PCR is aspecific primer binding of the DNA and giving unexpected products. So what can you do to reduce this mispriming? Do a nested PCR. With nested PCR, we won't stop after doing a regular PCR. We use a second primer set that binds just within the product of the first primer set. This increases the specificity and mispriming is reduced. How does nested PCR work? Primer design for nested PCR involves designing two primer sets, one for the outer fragment and one for the inner fragment. The first primer set binds to the outer pair of primer binding sites. This is like a normal PCR reaction. The second primer set binds to the first PCR product. The binding sites for the second primer set is between the first primer binding sites. The final PCR product will be only the target DNA amplified after the second PCR run. When performing a nested PCR, you always should take controls. The controls for nested PCR are the same controls for a normal PCR. A positive control with DNA to check the polymerase activity, and a negative control without DNA to check for contamination. So, nested PCR is used in situations in which it is necessary to increase the sensitivity and specificity of PCR. For example, detection of virus DNA, like hepatitis, with microbiological research, or with forensic research, like DNA fingerprinting.
Okay, so that's the principle and the application of the nested PCR.
What is an
And then the multiplex PCR. So in multiplex PCR, that means two or more primer sets designed for amplification of different targets are included in the same PCR reaction. Using this technique, more than one target sequence in clinical specimen can be amplified in a single tube. So this technique can save time and effort, and also the primer used in the multiplex reaction must be selected carefully to have similar annealing temperatures and must be not complementary to each other. And the amplicon size should be different enough to form distinct bands when visualized by gel electrophoresis. And the multiplex PCR can be designed in either single or template PCR reaction, okay, that use several sets of primer to amplify specific region within a template, or multiple template PCR reaction, which means you use multiple template and the several primer set in the same reaction tube. Okay. And the multiplex PCR is more complicated to develop, right? And often is less sensitive than the single primer set PCR. And the advantage of multiplex PCR is that the set of primer that can be used as an internal control, all right, so that we can eliminate the possibility of false positive, negative, and that also can save the costly polymerase, all right, because the tech polymerase is very expensive, and so that can save some money if you use a multiplex PCR. So in this picture, I show you that you can have the different sample, all right, amplify the same region, but you have different primer, all right, the label with different color and then with different size, and then you can visualize from the aqueous gel. You can see the different size or different fluorescence color that can be detected by real-time PCR with different color, all right, different result. Or you can have a different set of primer designed for one single template, okay? And in the laboratory, multiplex PCR normally can be used, and so that you can design a set of primer as the control, internal control, all right? And the other one be like experimental, so that you can measure the difference, all right, from the different sample with the same internal control. That's a multiplex PCR. And the digital PCR, all right? So we know the PCR exponentially amplify the nucleic acid, and the number of amplicon cycle and the amount of amplicon allow the quantitation of the starting quantity of target nucleic acid. However, many factors complicate the calculation. So that will create uncertainty and inaccuracy, especially when the starting concentration is very low. Then the digital PCR can overcome this difficulty by transforming the exponential data from conventional PCR to digital signal, so that they simplify, just indicate whether or not amplification occurs. So they will only give you positive, negative, 1, 0, that's it. So the digital PCR is accomplished by capturing or isolating each individual nucleic acid molecule presented in the sample within many chambers, okay? And so now that localize and concentrate the amplification product to detectable level. Then after the PCR quantification, a count of the area containing the PCR product is a direct measure of the absolute quantity of nucleic acid in the sample. Okay. So the application of digital PCR, so basically that would be the detection and quantification of low level of pathogen sequences, expression of the rare genetic sequences in single cells. All right. So these can be used, right, at digital PCR, because they only give you this positive, negative result. They don't give you the amount. Okay, and that's the digital PCR. So now here's just the video to show you the digital PCR.
One of the greatest challenges in molecular biology is the detection of a rare allele or mutant sequence lost in a dense background of wild-type sequences. This situation is often encountered in oncology, for example, when trying to detect tumor-derived mutant sequences present in very small amounts in liquid biopsies. In this animation, the mutant sequence is colored in red, and the wild type is colored in green. Finding the mutant sequence is a typical needle in a haystack problem. When using standard, real-time quantitative PCR, the mixture of DNA molecules is amplified in bulk in a reaction mixture containing PCR reagents, including attack polymerase, PCR primers, and 2 types of probes, each specific for the wild type or the mutant sequence respectively.
So in this example, they put 2 type of probe, right? So this is an example of the multiplex PCR, the application multiplex PCR, okay?
The wild-type probe is labeled with a green fluorophore. The mutant probe is labeled with a red fluorophore. During the PCR amplification, the probes specific for the mutant and wild-type sequences anneal to their respective targets and release a fluorescent signal. However, in a bulk reaction, the signal of the mutant target is in competition with the signal of the wild-type one. And when the mutant sequence is present at a very low fraction, it may not even be detected. To overcome this problem, digital PCR relies on a basic principle: partitioning the sample prior to PCR amplification in order to isolate individual DNA molecules in different compartments. Today, several techniques are employed to achieve the partitioning. This is either done in solid microchambers or done in an emulsion of microdroplets. When the sample is partitioned, all DNA molecules are randomly distributed into a large number of partitions, such that only one or a limited number of DNA molecules end up in each partition. Even if mutant and wild-type sequences end up in the same droplet, the competition between the two is greatly reduced. During the PCR amplification, a fluorescent signature is generated within each partition according to its DNA content. One targeted DNA molecule is sufficient to generate a fluorescent signal in a given partition. Finally, the concentration of wild-type and mutant sequences in the sample is precisely estimated by counting the number of each type of partitions, classified according to the measured fluorescence levels: the non-fluorescent negatives, the greens, the reds, and the double positives. Digital PCR also enables the simultaneous detection of multiple targets using a multiplex assay. Rigorously, A formula based on the Poisson law converts the countered negative and positive partitions into the target concentration of both the wild type and the mutant sequences. Digital PCR is a powerful tool for the detection of rare events, but can also benefit other assays, such as copy number variation, by being able to precisely measure less than two-fold differences in copy number, or absolute quantification of nucleic acids, since this technique does not have to rely on comparison with pre-calibrated standards. Digital PCR is also a great tool for genotyping single cells. All those assays are currently applied to a wide range of applications, such as oncology, infectious diseases, environmental testing, prenatal diagnosis, organ transplant, etc.
So that's the principle and application of digital PCR. Okay. So the parameters like the FACT PCR, I think we should discuss this previously, but I don't know, maybe misplaced in here. So it should be before we discuss the modification PCR. It's okay, we can still discuss here. So the parameter that can affect the PCR, the first one is the size of a PCR product. Okay, the first step to design a primer is determine the target region that is for PCR amplification. Normally, the target sequence to be amplified should be kept below 3 kilobase. Okay, because fragments up to 10 kilobase can be amplified by standard PCR technique, but the longer the fragment, the less efficient the amplification. And furthermore, it is more difficult to obtain consistent and accurate results in the longer amplification. In the meantime, that the polymerase can have the opportunity to introduce the mismatch error. Okay. Because the longer PCR fragment, you need a more specific DNA polymerase to perform such a job, because in the original tech DNA polymerase, they might introduce a lot of error when you try to amplify more than 3 kilobase long. But nowadays they did engineer to create some specific tech polymerase that can increase the accuracy and the length of the primer. Okay, once the target region is determined, then the next issue is the length of the primers. If the primers are too short, they might hybridize to the non-target sites and give undesired amplification product. Okay. So that be the this is just an example. If the length of primer too short, if it's 8 mer, okay, so that means only a nucleotide, and each nucleotide position you have 4 different possibilities, right? So that means 4 to the power of 6, 4 to the power of 8. Then that means that every 4 to the power of a nucleotide, then this primer will find their target. If you design a primer with 17 mers, that's 17 nucleotides, so that means every 4 to the power of 17 bases, then you will find the exact same sequence. All right, so that will reduce the non-specific binding. If you compare just 8 nucleotide long primer to the 17 nucleotide long primer, these will have more non-specific binding than this one. So the length of primer is also important, okay, to increase the specificity. And annealing temperature, as I mentioned earlier, so annealing temperature, if it's too high, then no hybridization will take place, okay, because it's a very high stringency condition. If it's too low, then that will increase the non-specific binding. So it's a low stringency condition. And so that's why it always recommend to have like a 2 degrees below the melting temperature to start to begin with, and then you can adjust the melting temperature based on your result. Okay, so these are 3 important parameters that can affect the PCR results. Now, the most important one, okay, it's called a real-time PCR, which is a quantitative PCR. Okay, it's a technology for the quantification of nucleic acid at the real time. And because it quantifies the amplification of a DNA during the reaction, okay? So they can detect the accumulation of amplicons after each thermal cycle in the real time. And the real time that implies the data collection and analysis occur as the reaction proceeds. And the initial concentration of target sequence can be determined by comparison with the amplification kinetic between the target and the internal control with non-copy numbers. All right, so that's why now when many agencies they perform this the QRT PCR, the qPCR, the QRT PCR, the real-time reverse transcriptase PCR to detect the presence of the SARS-CoV-2 virus. Because of this real-time PCR, they can even detect the presence, not only the presence of the RNA, but also they can give you the amount of the initial concentration, so that can determine how much virus in the human body. Okay, and so the QRT-PCR is a very powerful technique nowadays, especially during this pandemic time. And also QRT-PCR can be used to determine the gene expression analysis. Okay. And can be used for the quantification, genotyping of pathogen, gene expression, and also epigenetic analysis. So many powerful application through the QRT-PCR. And this is the typical curve that you will see after the PCR reaction. Okay, so during the PCR reaction, as I mentioned, that they can monitor the amount of the amplicon in the reaction after each cycle. So here the x-axis, that's the number of cycle, and the y-axis is the amount of the product. And during this evaluation, okay, during this process, you can see the 1, 2, 3, 4, 5, 6, 7 different sample is running. All right, these 7 sample are running during the QRT-PCR, and you see this red line here. That's the threshold cycle. All right, so that means at last cycle, that specific reaction, their product, amplicon product, all right, they detect the machine detect the presence of product above the background, all right, above the background. So that's the place where they call the threshold cycle. Okay, this is the fluorescence signal they detect from the machine. And so for the first sample, as you can see, the first 8 cycle, the machine did not detect anything because all the signal are below the background. Then until this cycle 11, suddenly they increase. So actually, starting cycle 9, the machine slowly detect the signal, all right? And then until cycle 24, that's the plateau, the maximum. And then, of course, the machine, they can automatically detect where would be the threshold cycle, which means where would be the place where the signal is above the background. Okay, and then you will see the number when you later on when you look at the data, right? So that this is the threshold cycle. And then for these 7 different sample, you can compare these threshold cycle, all right? So this is so-called a CT value, threshold cycle value. So for example, the first sample will be like a CT value is 11, and then the number seven sample, the CT value is 32. At current time, when they evaluate whether your PCR results positive, negative with the COVID-19, all right, so based on the CT value, all right. So for example, this one, the first sample here, the CT value is 11. So the CT value is very low, so that indicate the initial concentration of the sample is very high. So that means if you use these in the COVID-19, that means this patient has the COVID-19 virus because the concentration is very high. That's why the CT value is very low. All right. When you have a very high concentration, that means you only need very little amount of cycle to amplify your target to reach that to reach the point where the fluorescent signal is above the background, right? And then for this curve, that means this patient might not have the COVID-19 because that would be almost nothing. When you have the threshold cycle above 30, 32, that's a very, very lower initial concentration. So that means that person might not have the COVID-19 virus. If you use these, apply this in the COVID-19 testing, and this is how they evaluate the data. Okay, and then from this curve, you can see for the first curve here, they can reach the plateau, right, at probably the cycle 25, all right, and then the number 7 reach the plateau on about cycle 38. If you perform the conventional PCR, and then you have this 40 cycle amplification, you would not be able to distinguish the difference between the number 1 and number 7 curve, right? Because for the conventional classical PCR, you only evaluate the end product, which means you only look at the result at the end of the PCR reaction, because all the sample reach the plateau at the end of the amplification. So you will not be able to differentiate the difference between number 1 and number 7. Only the real-time PCR, so you'll be able to see the difference. All right, so that's the major difference between the conventional PCR and the regular the QRT PCR. So that's the comparison, right? The between the qPCR and the conventional PCR. And because this PCR, qPCR, as you can see, different color, because they have to monitor the presence of sample at the real time, so that they need to have the fluorescence signal to be detected, right, by the machine. So that we need a different fluorescence dye to be added to the sample, so that you'll be able to measure, to detect, to monitor the sample. So there are 2 different type of measurement. One is non-specific detection dye. Okay, so non-specific detection, that means the dye that you use, they can bind the DNA non-specifically. Okay, non-specific. So like acidity bromide, cyber green. So these 2 dyes, they bind DNA non-specifically. Okay, and they will measure the amount of the, because acidity bromide, they bind a double-stranded DNA. All right, and as we learned that every cycle, in the end of a cycle, you have a double helix DNA. And then for the first cycle, for the 0 cycle, you have 1 double helix DNA. 1 cycle, you have 2 double helix DNA. And 3 cycle, you have 8 double helix DNA. So then as you increase the cycle, the amount of double helix DNA increase. So the more double helix DNA you have, then the more ethylene bromide will bind. And so when you more ethylene bromide bind to your double helix DNA, then the machine will detect more signal. All right, so that's how they determine the amount of the DNA. So that's the DNA binding dye chemistry. Okay. It's a DNA specific, but they have non-specific target. Okay. And then another type of the DNA analysis in qPCR will be hybridization probe. So this will be the fluorophore link probe for specific DNA, PCR product detection. This will be very specific at the specific to your, specific to your target. All right. So they have an extra probe that labeled probe to determine the specific target amplification. Because for a non-specific detection, they detect any DNA, double helix DNA, the presence the double helix DNA, even it's a mispriming, even it's a mispriming PCR product, they can still give you the signal. But for this hybridization probe, that'll be very specific target to your defined region. Okay, so that'll be TechMan, molecular beacon. That we will also discuss later. So the cyber green, all right, cyber green, and also the EVA green. The EVA green is the third generation of DNA binding dye. Okay, they bind any double-stranded DNA. All right, they can bind any double-stranded DNA. So during the amplification, the extension stage, right, the more you have, then the more dye will bind, and then you will detect the signal. And because these kind of dye, they bind the DNA non-specifically, all right? So that means even if the PCR reaction might have not mispriming and the mismatched product, or even the non-specific product, primer, dimer, they can be formed during the reaction, and then as long as you have the double-stranded product, the cyber green, ethylene bromide, EVA green, they will all bind and give you the signal. So to distinguish the difference, okay, we have to assume this PCR product should have different melting temperature, all right? Should have a different melting temperature. So for this kind of non-specific detection, for ethylene bromide, cyber green, normally you have to perform the melting curve analysis to check the specificity of the amplified fragments. Okay. So basically, the melting curve analysis that will show the temperature of DNA denaturation. Okay, should have only one single peak, right? So that means one single peak. That means the product is only a single product that to your target. If you see a different peak, that means that could come from the primer dimer or the non-specific binding. Okay. So that's a melting curve analysis. Normally, you have to perform this to ensure that your PCR reaction is correct. Okay, that's the for the qPCR. And the cyber green and the ethylene bromide are very cheap, but they have some limitation. The cyber green, so for example, they have a limited dye stability and dye-dependent PCR inhibition. All right. So that's a cyber green. And also sometimes the cybergreen, when you have a high concentration cybergreen, that can inhibit the PCR reaction and also promote the mispriming. Okay. And the low cybergreen concentration can compromise the PCR signal strength. Also, make the DNA melting curve data unreliable because the dye redistribution phenomenon. Okay, that's the problem when you use a cybergreen and EVA green. So it's basically has less inhibitory to PCR than the cybergreen, can be used under the saturating condition to generate greater fluorescence signal. Okay. So that's the EVA green, that's the third generation. So these are some DNA dye that can be used in the qPCR. So now let's watch the overview of qPCR reaction.
In contrast to traditional PCR, where the presence or absence of a DNA sequence is analyzed upon reaction completion, quantitative PCR, or qPCR, monitors DNA amplification.
So for the traditional PCR, right? So basically for first, let's say for first 30 cycles, you will not be able to monitor the first 30 cycle. You only evaluate the end result. So when you evaluate the end result, everything you look similar, right? Because you cannot see the difference, right? You did not monitor the process. For the qPCR, they monitor the process so they can distinguish the differences.
As the reaction progresses. In qPCR assays, reaction progression is monitored by an increase in fluorescent signal. The fluorophore can be a double-stranded DNA binding dye, most commonly cybergreen 1, or covalently attached to a target-specific probe.
So this is target-specific probe, which we will discuss later, is a probe chemistry. And one of the commonly used ones called TaqMan.
As in TaqMan assays. In cyber assays, the dye fluoresces when bound to double-stranded DNA, but displays weak fluorescence in the presence of single-stranded DNA. This results in an increased fluorescent signal as the reaction progresses and double-stranded DNA is formed. The most common probe-based assay is a 5' nuclease assay, known as TaqMan. In TaqMan assays, the 5' flap endonuclease domain of Taq DNA polymerase.
For a TaqMan probe, okay, so they have designed not only a pair of a primer, but also a third probe here. And the probe must be within the range of these two primer. And the probe must be labeled, right? Must be labeled so that they can bind to the specific region, which is your target. As they proceed, they will release the signal.
Cleaves a target-specific probe, separating a fluorophore on the 5' end from a quencher on the 3' end, resulting in an amplification-dependent increase in fluorescence. A thermocycler, coupled with a fluorimeter, modulates the reaction temperature during DNA amplification, while also monitoring the fluorescence signal at each PCR cycle, generating a real-time qPCR curve. There are 4 distinct phases within the amplification curve: lag, exponential, linear, and plateau. During the lag phase, amplification occurs. The signal is not strong enough to be detected above background. The exponential phase is used for quantification because it produces a fluorescent signal measurable over background and contains an abundance of reaction components, permitting a detectable doubling event at every PCR cycle. When the fluorescent qPCR signal is detectable over the background fluorescence, a quantification cycle, or CQ value, can be determined. CQ values can be used to evaluate relative target abundance between 2 or more samples, or to calculate absolute target quantities in reference to an appropriate standard curve. qPCR can also be modified to detect and quantify RNA. Interpretation of qPCR data requires careful experimental design and analysis.
So that's the overview of the qPCR. And during this presentation, he mentioned CQ. Remember, it's not CQ, it's CT value, okay? The threshold cycle. That's the signal when the machine can detect above the background. And that's also the value nowadays the whole world use to determine the presence of the COVID-19 virus or not. That's CT value. Okay. Oh, here, let's watch this, how to use the reverse transcription PCR to detect the COVID-19.
Using reverse transcription polymerase chain reaction, RT-PCR, in COVID-19 testing. COVID-19 testing usually begins with a swab of the nose or throat. Saliva, mucus, or fluid from a patient's lungs can also be used. The swab of a person with COVID-19 will contain a mixture of human cells, virus particles, and other microbes. Like all living things, the human cell has DNA as the genetic material that passes on information from one generation to the next. The DNA molecule is made up of two strands that look like a twisted ladder or double helix. However, the COVID-19 virus, SARS-CoV-2, and many other viruses, including HIV, have RNA as their genetic material, genome. RNA is chemically very similar to DNA, but has only a single strand. The virus RNA is surrounded by a nucleocapsid protein within the virus envelope. Other proteins are embedded in the envelope itself. The SARS-CoV-2 genome contains genes, blue arrows, that carry the direct.
So the first step, as I mentioned, you have to sequence the whole genome, right? It's about 30,000 kilobase long. And then we have S protein here. That's the one, the major one that recognize the receptor so that the virus can enter the cell.
For making these and other proteins that are needed to replicate the virus inside the human cell. The objective of COVID-19 testing is to identify part of the viral genome in the patient sample. This is usually the N gene, which carries directions for making the nucleocapsid protein. There's not enough viral RNA to detect directly in the patient sample, so a process called reverse transcription polymerase chain reaction, RT-PCR, amplifies many copies of a segment of the N gene. Short, single-stranded pieces of DNA, called primers, recognize unique RNA sequences within the viral genome that bracket the target region of the N gene. After the first primer binds, an enzyme called reverse transcriptase extends, synthesizes, a single-stranded DNA copy of the viral RNA, known as complementary DNA, or cDNA. After the RNA is removed, the second primer binds to the other side of the single-stranded cDNA. Then, a second enzyme, taq-DNA polymerase, extends a second strand to produce the double-stranded DNA copy of the target region of the viral RNA. This DNA copy then undergoes successive rounds, cycles of amplification, during which the DNA separates, denatures into single strands, both primers bind, anneal, to their target sequences, taq polymerase extends, synthesizes a new DNA strand, and so on. The number of copies of the target region of the viral genome doubles with each cycle. After 30 cycles, up to 1 billion DNA copies of the viral RNA are produced by PCR. In practice, the virus is typically detected with 30 to 45 cycles of PCR. Adding a fluorescent probe allows the amount of target DNA.
So now, if you have the fluorescent probe, then that'll be the qPCR. And since it's RT-PCR, so the whole process is called qRT-PCR.
DNA to be detected in real time and quantified after each cycle of PCR. This graph shows the detection of 200. Using reverse transcript each cycle of PCR, this graph shows the detection of 220 and only 2 virus RNA molecules in a controlled study. Results.
So as you can see the curve here, they didn't show here. The blue one, they show the CT value earlier, right? And then this one, the green one, that'll be the control. The earlier you show them, that indicate the more virus you have, okay?
20, and only 2 viral.
So as you can see, the earlier you can see the curve, that means you have a higher concentration of virus, right? And the less you have, then you will see the curve to show up later, and then higher CT value, okay? And this is low CT value. Low CT value, that means you have a high amount of a virus.
RNA molecules in a controlled study. Results from swabs vary. A negative result is also shown. See these DNA Learning Center animations for a more detailed look at the PCR process.
So that's the procedure to perform the qRT-PCR to determine the presence of the virus in the human body. Okay, and that's the first one we talk about. We discussed these the cybergreen acidine bromine non-specific DNA binding probe, and now the second detection method will be the fluoro labeled oligonucleotide for the qPCR. Okay. So there are two types of the fluoro. Okay, two types. One is the donor reporter and also the receptor quencher. Okay, and the fluoro used in qPCR can be the primer probe, right? Probes or the analog of nucleic acid, right? The primer probe, that means they design the primer. Also, the primer can be used as a probe. Okay, that's called a primer probe. And the probe, that means you design extra probe on top of the 2 primers. And analog nucleic acid, that's directly you use the modified nucleic acid. Okay, so these are the different type of the chemistry they use. Okay, for the primer probes. Okay, the primer probe are oligonucleotide that combine a primer and the probe in a single molecule. Therefore, the dye does not have to add it directly to the reaction mixture because the primer already attached the fluoro. Okay, and these assays are relatively inexpensive to run and also fairly straightforward to design. The fluorescence emitted from the primer probe is detected and measured during the denaturation or expansion phases of the qPCR, depending on the type of primer probe used, and the use of these primer probe can lead to the amplification on specific product or dimer primer during the PCR reaction. Therefore, melting curve analysis to determine the efficiency of the reaction is always recommended for this application. And the primer probe can be classified into 3 groups. It can be a hairpin, cyclicone, angular primer probes, right? These 3 different groups. And this is hairpin primer probe. So hairpin primer probe are single-stranded oligonucleotide with 4 features. Okay, one would be the hairpin secondary structure, right? Like this hairpin secondary structure, so that the loop of the structure specifically bind to the target DNA. Okay, the loop, this loop here that can bind to the target DNA, and the probe contains a short tail sequence of 6 nucleotides equals CG, the 6 nucleotide that complementary to the 3' end sequence. Okay, and also the different type of the hairpin primer probe that include scorpion, amplifluor, light upon extension, LUX. Right, these 3 different kind of hairpin primer probe. So they have this different kind of design, right? So we are not going to discuss this in detail, just to know that there are different type of the help in a primer probe. And then the second type of the probe will be a cyclicone probe, which has a long primer probe, which is complementary to the target DNA sequence, and also a short modified oligo attached through the 5' to 5' and that will bind to a 6 to 8 nucleotide at the reporter containing the modified 3' end of the primer probe. And also they have a quencher, right? The quencher basically is an assignment based at the 5' position in the primer probe sequence. And so the binding, they form this the cyclic structure with 2 or 3' end. So that's the cyclicone probe. And the angular the primer probe, basically they have a DNA sequence identical to the target, and which is bound to a reverse primer through a hex ethylene glycol linker, HEG, all right? And it has an acceptor fluorescence moiety at its 5' end, okay? And the cyber gold DNA intercollecting dye is employed in the assay as a donor fluorescent moiety, all right? And in the solution, the primer probe does not emit the fluorescence because there is no donor fluorescent moiety close enough for the signal. When the angular primer probe binds to the target DNA during the annealing step, the DNA polymerase starts the extension of the 3' end reverse primer, and then during the denaturation phase, the specific sequence of the probe binds to the complementary region of newly amplified DNA, producing a double-stranded DNA fragment, and therefore the cyber gold dye can be intercollected to generate the fluorescence. And then the emitted fluorescence can be measured during the denaturation step in each cycle. That's angular primer probe. Okay, and then for the probe-based chemistry, that means you have to design extra third oligonucleotide as a probe, and then you label the probe with the fluorescence. Okay, you have this extra fluorescence probe, and this is complementary to a target sequence and in between the two PCR primers. And there are two types of probe, the hydrolysis and the hybridization probe. Okay. The TaqMan probe is a hydrolysis probe, and then the molecular beacon will be the hybridization probe. For the hydrolysis probe, so basically it's rely on the 5' to 3', this 5' to 3' exonuclease activity, okay, of the TaqDNA polymerase. They can degrade these bound probes because this probe, TaqMan probe, they have reporter dye and quencher, right? When you have this probe bind to the target, right, there's no signal. But the Taq polymerase, when they perform the DNA synthesis, they will have the 5' to 3' exonuclease activity. They digest this probe, and then they can release the signal so that you can detect the signal. Okay, that's a hydrolysis probe. So the fluorescence is measured at the end of the extension phase. So it is proportional to the amount of amplified specific product. That's the TaqMan probe, and this molecular beacon probe is the hybridization probe. The fluorescence emitted by binding the hybridization probe can be measured during the either the annealing or the extension phase. They can allow the amplified fragment to be analyzed by performing the melting curve, and this is the main advantage over the hydrolysis probe. The amount of fluorescent signal detected is directly proportional to the amount of a target amplified sequence during the qPCR reaction. Okay, so that's the typical hybridization probe are the molecular beacon probe or the high probes. And the high probe is known as a FRED probe, F-R-E-D, FRED probe, that's high probe, FRED probe. They have a pair of oligonucleotide binding to the adjacent target DNA sequence, and the one probe carry a reporter, all right? A reporter at 3' end, and the other probe contain the quencher at 5' end. And also the phosphate group attached to the 3' end. This is a phosphate group at 3' end. And the sequence of the probe are designed to hybridize to the target DNA sequence in the head-to-tail orientation so that the two fluorophores are in close proximity, all right? So they are in close proximity so they can, the signal will not detect, all right? During the annealing stage, all right, the probe are adjacent to the bond. The quencher emits fluorescence because it has been previously excited by the energy released from the reporter. The advantage of this probe is that their design and synthesis is quick and easy. They can be used in the multiplex format for pathogen detection, right? Viral, bacterial load quantification, microarray validation, all right? So that's the FRED. And molecular beacon probe are single-stranded helping see these single-stranded helping shape oligonucleotide probe. So the probe usually are divided into two parts. You have a loop, right? You have a loop, which is a fragment, 18 to 30 base pair complementary to the target DNA sequence, and also you have a stand, right? A stand here is formed by the two complementary sequence of 5 to 7 base pair. And then the 5' end of the probe has a fluorescence reporter, and the non-fluorescence quencher is in this 3' end, because they can absorb the emitted fluorescence from the reporter when the molecular beacon probe is closed to form. During the annealing phase, the probe will be unfolded, right? Will be unfolded, and then they will bind to the target. They bind to the target, and then the fluorescence is emitted because the reporter is no longer quenched. So the fluorescence signal is proportional to the amount of amplified PCR product. If the molecular beacon probe and target DNA sequence are not perfectly complementary, there will be no emission of fluorescence, okay? Because the hairpin structure prevail over the hybridization. So that's a molecular beacon. So let's watch the video about this. The probe-based qPCR.
Real-time PCR, also known as quantitative or qPCR, determines the actual amount of PCR product present at a given cycle. By using a fluorescence report in the PCR reaction, this process allows you to measure DNA generation in the qPCR assay. In a probe-based qPCR reaction, you have your template, which contains the target sequence that you are interested in. You also need primers, dNTPs, and DNA polymerase of your choosing. In addition, you will need a probe labeled with both a reporter molecule and quencher molecule. Typically, probes are sold as separate, custom products because they are specific to the target sequence. Denaturation is the first step in the PCR reaction. The thermocycler heats up to roughly 95 degrees Celsius, which causes the double-stranded DNA helix to melt open into two single-stranded DNA templates. During the annealing step, the temperature cools between 45 to 65 degrees Celsius. Probes, labeled with both a reporter molecule and a quencher molecule, and sequence-specific primers, anneal to the single-stranded DNA template. During this cycle, DNA polymerase attaches to the prime template and begins to incorporate complementary nucleotides. Finally, during extension, the temperature slightly rises to 65 to 75 degrees Celsius. During this phase, DNA polymerase extends the sequence-specific primer with the incorporation of nucleotides that are complementary to the DNA template. The DNA polymerase then displaces the reporter molecule from the probe, resulting in fluorescence. The fluorescence accumulates as cycling of PCR continues and is measured at the end of each PCR cycle. The intensity of the fluorescence generated by the reporter molecule above background level, the CQ value, is measured and used to quantitate the amount of newly generated double-stranded DNA strands. After repeating the denaturation, annealing, and extension cycles approximately 35 to 40 times, you are ready to begin analysis. The CQ values can be used to quantitate starting amounts of DNA, establish a standard curve for gene expression studies, or other analysis. Probe-based qPCR is a great option for specific hybridization, no false positives, and amplification of two distinct sequences in one reaction tube. Instrument compatibility is critical.
So that's the probe-based qPCR. And then nucleic acid analogs, right? So we have different types of nucleic acid analog. They are compounds that are analog, which means the structure is similar to the naturally occurring DNA and RNA. So an analog may have an alteration in its phosphate backbone, pentose sugar, or nitrogenous base. Normally, the analogs incorporate all of the advantage of native DNA but are more stable in biological fluids and have increased affinity for complementary nucleic acid target. Right here, just examples some typical analog. So like the PNA, that's the peptide nucleic acid, right? PNA, like this peptide nucleic acid. See the nitrogenous base. And the RNA, that's a log nucleic acid here. See the structure here, the log nucleic acid. And the ZNA, which is here, the ZNA, and that's called a zip nucleic acid, all right? And they can compare this normal naturally occurred DNA structure, right? So you can see the structure very similar, okay? So the PNA, basically the sugar phosphate backbone, the PNA has been replaced by the peptide of the N2 amino acid or glycine unit, right? So this part, sugar phosphate, has been replaced by this peptide of the N2 amino acid or glycine. And the PNA are electrically neutral DNA analog. PNA hybridized to complementary oligonucleotide. Okay, based on this nitrogen base, they have this complementary sequence they can bind, all right? And RNA, right? RNA, that's the DNA RNA sequence in A conformation that contain one or more modified nucleotide. And then they have this bicyclic ring, all right, through the methylene bridge between the 2' oxygen and the 4' carbon. And the RNA nucleotide can be introduced into the primer probe, and the probe, like the molecular beacon and TrackMan probe. And RNA are resistant to degradation by nuclease. Okay. And the ZNA are a novel type of synthetic modified oligonucleotide, right? The ZNA, because they have this conjugate in a cation in a moiety, Z unit, right? This Z unit. And these modification, they can increase their affinity for the target during the annealing phase. So that's the commonly seen the analog and the detection system. So the thermocycler with the fluorescence detection, all right? So that would be a specialized the real-time PCR, and because they combine with this fluorescent detection system, so therefore the the real-time PCR equipment is very expensive. Each one at least like a $50,000, very expensive. Okay, it's more than the cheapest BMW, the 3 Series. Okay, the qPCR machine is very expensive. And here's the advantage to use the PCR. Okay, and here's the limitation, PCR reaction. Okay, so now the limitation of PCR amplification in molecular diagnosis is the requirement of a thermocycler, right? The thermocycler, the first step, 90 degree, right, to separate the two DNA strand, the template DNA strand. And thermocyclers are usually costly and not portable, right? So for real-time PCR, as I say, each of the real-time PCR is $50,000, and the regular conventional PCR, which does the conventional regular PCR does not have the fluorescence detection. That cheap one would be like a $5,000, okay? But you cannot use that for real-time. For real-time, at least 50,000. So the limitation for PCR application, number one would be that the equipment, very expensive, all right? And so it's not possible to be used in the point of care testing, all right? A wide variety of isothermal amplification techniques have been developed without using the thermocycler. The non-PCR based method has been developed according to some new finding in molecular biology of DNA RNA synthesis, right? Because basically they have the modified or newly discovered enzyme that can be used, right? So it's isothermal amplification. And then the two different types of isothermal amplification. One is transcription-based amplification. That include the transcription-mediated amplification, that's called TMA, and also the nucleic acid sequence-based amplification, NASPA. Okay, both are isothermal amplification reaction, and it's mimicking retroviral RNA amplification. And both are specific for target RNA sequence, have been gaining popularity because they have been shown to have a wide range of application for pathogen detection in clinical, environmental, and the food sample. And both the TMA and NASPA, all right, they have the advantage of not requiring the thermal cycling. And these two techniques use the function of RNA polymerase to make RNA from a promoter engineered in the primer region, and the reverse transcriptase to produce DNA from the RNA template. For the TMA, the reverse transcriptase cell degrade the initial RNA sample as it synthesizes the complementary DNA. Okay. And this is for the NASPA, all right? NASPA, the RNA amplification technology, basically you introduce the third enzymatic activity, which is called RNSH. RNSH basically to remove the RNA from the cDNA RNA hybrid, okay, without a heat denaturing step. During the reaction, the forward primer hybridized to a target RNA present in the sample. Then the enzyme reverse transcriptase and the RNA-SH along with the reverse primer, then produce the double-stranded DNA with the target sequence and T7 promoter. And this is just the procedure. So initially you have the RNA template, then you have the antisense primer, okay, with the presence of a reverse transcriptase, all right? So you synthesize these, and then you have this RNA DNA hybrid. Then the RNA-SH will degrade the RNA and give you these DNA, single-stranded DNA. And then you have the sense primer with T7 promoter. Then you can synthesize this double-stranded DNA. Okay, this is an SBA. And then another one is called a signal-mediated amplification for RNA technology, called SMRT SMART. Okay, the SMART is based on the formation of a three-way junction structure, like here's three-way junction structure. And then you need the two single-stranded oligonucleotide probe. Each of the probe, you can see they have a region, right? They can bind to the template, right? So they have the region hybridized to the target, right? And also they have another shorter region that hybridized to other probe here, right? So it's one probe, first probe, second probe. They first of all have the region that can hybridize to the target, and also they have a shorter region that can hybridize to each other, complementary to each other. Okay, they can near to each other. So you have this three-way junction. And also another long probe also contains a T7 RNA promoter and the transcription template. Okay, so after this formation of a three-way junction, right, the DNA polymerase, in this case for the SMART technology, you have a specific DNA polymerase. It's called a BST DNA polymerase. So the BST DNA polymerase, then they can extend this. If after they form this three-way junction, all right, 5', 3', 5', 3', and T7 promoter, so they can extend this, all right? Extend this, and you have this template here. So they can form this extension here, right, from the T7 RNA polymerase promoter. And they allow the T7 RNA polymerase to generate another copy of this RNA amplicon, right? So that's only produced when the specific target is present to allow, if you have this target, then they can form the three-way junction, then they can synthesize here RNA. Okay, then each RNA amplicon itself be amplified by binding to a second template oligonucleotide, right? So here you amplify, right? Amplify, and then you bind to the second template oligonucleotide, and then extend it by DNA polymerase to generate double-stranded promoter. And this will lead to the transcription that increase the RNA amplicon. The RNA amplicon can be detected by enzyme-linked oligosorbent assay or in real time. So basically, the method actually rely on the signal amplification, but does not require the thermal cycling. Okay, so that's a SMART technology. And then the strand displacement amplification is called SDA, right? Use the activity of the restriction endonuclease and also an exonuclease deficient strand displacing DNA polymerase to generate copies of a target DNA sequence. The SDA combines the ability of restriction enzyme to nick the unmodified strand, right? Unmodified strand of the target DNA, and then action of an exonuclease deficient DNA polymerase to extend the 3' end at the nick and displace the downstream DNA strand. The displaced strand serves as a template for an antisense reaction and vice versa, resulting in an exponential amplification of the target DNA. So basically, after the initial denaturation, to separate the double strand of DNA, the extension of SDA primer containing the restriction enzyme, right? And then their displacement by extension of a flanking bumper primers produce double-stranded target sequence. Okay, so that's the SDA displacement amplification. And rolling circle amplification use the strand displacing DNA polymerase to continuously amplify a circular DNA template at constant low temperature, producing a long DNA molecule with tandem repeat of the circular template. So that's an RCA amplification. And the loop mediated isothermal amplification is called LAMP, which is the most commonly used technology. They use a set of 4. You need the 4 specifically designed primer, and these 4 primer, they recognize 6 regions on the target DNA, and the DNA polymerase with strand displacement activity to amplify a target 1 billion times in less than 1 hour at a temperature around 60 degrees. And the amplification produce the stem loop DNA structure with several inverted repeat of target and the cauliflower-like structure. Okay, like this cauliflower structure. Later we will watch a video, okay, to show you this LAMP technique. So let's watch this LAMP.
When designing a LAMP experiment, 4 to 6 primers are required.
So you need the 4 primer, all right? And then they recognize 6, they have 6 binding site. So forward outer primer, inner primer, reverse outer primer, reverse inner primer. Okay, outer primer, inner primer. Okay, watch carefully.
Specific to identified regions of target DNA.
Here.
When experiment are required. Specific to identified region.
So here, so we have the 4 primer, right? We have the 2 forward to outer primer, the forward and reverse outer primer. So you can see the F3 primer bind here, B3 primer bind here, all right? Bind here and here, the outer primer. Then we have a set of the inner primer. They recognize 2 regions here, F1, F2. See F1, F2, right? F1 bind here, F2 will bind here. B1, B2, B1 bind here, B2 bind here. Okay, these are their target site, okay?
Of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the.
Okay, the strand displacement, right? So that begin from the F2, right, bind to the complementary sequence. Okay, the inner primer.
Are primers. And a strand displacing DNA polymerase extends the primer and separates the target DNA duplex.
So that they duplicate this strand, right? duplicate this strand.
The first product is then.
And then F1, the first product. Remember, there's another sequence here. This will form a complementary sequence with F1 here.
Displaced by synthesis, initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion.
So when the outer strand started, then they will be displaced. Then this synthesized DNA, they will form this loop, right? From this loop.
Of a reverse complementary sequence in the inner primer sequence.
Okay.
This annealing and displacement cycle repeats on the opposite end of the target sequence, and the resulting is a short dumbbell structure that forms the seed for exponential lamp amplification. This lamp dumbbell structure contains multiple sites for initiation of synthesis, from the 3' ends of the open loops and annealing sites for both the inner and loop primers. As amplification proceeds from these multiple sites, the products grow and form long concatenamers, each with more sites for initiation. The result is a rapid accumulation of double-stranded DNA and amplification byproducts that can be detected by a variety of methods.
So in this process, as you can see, you do not need to have the denaturation step. Okay, they use the strand displacement mechanism to perform this amplification. Okay, here is also another one. So this is template, right? Template, and then we have these 3 regions, the target region, and then you can design the 4 primers and begin with the inner primer here, all right? The inner primer, hybridization, then elongation, then the strand displacement, another primer come in, F3 come here, and this will be displaced, and then F1C, they form this complementary sequence here, and now FB2 amplify, displacement, right? Remove this piece, they form the this dumbbell structure, right here. You can find here. Like this. So they can amplify the target very quickly, right? Without a change of temperatures, LAMP technique. Okay, so that's the end of the chapter 9.